welcome to Cancer Wars episode 6, where we're going to be talking about genomic instability within cancer cells. So, the acquisition of these 6 hallmark capabilities of cancer during tumor progression presents a significant dilemma. These capabilities include sustaining proliferative signaling, evading growth suppressors, resisting cell death, enabling replicative immortality, inducing angiogenesis, and activating invasion and metastases. And most of these capabilities are acquired through changes in the genome of cancer cells. However, mutations in specific genes are typically rare due to the diligent maintenance of genomic integrity by various DNA monitoring and repair enzymes. These genome maintenance systems strive to keep DNA sequences pristine, and checkpoints during cell division help ensure chromosomal stability. Consequently, the multiple mutations present in tumor cell genomes are highly unlikely to occur within a human lifespan. However, despite the inherent stability mechanism, cancer still appears frequently in human populations, and this frequent occurrence has led to the hypothesis that cancer cells must acquire increased mutability to complete human progression within several decades. The malfunction of specific components of these genomic caretaker systems can increase this mutability. For example, the p53 tumor suppressor protein is a key member of these systems. In response to DNA damage, p53 can either halt the cell cycle to allow for DNA repair, or it can trigger apoptosis if the damage is too severe. Apoptosis is a form of programmed cell death that prevents proliferation of damaged cells, and the loss of p53 function, which is common in most human cancers, highlights its crucial role in maintaining genomic integrity. So imagine p53 as a vigilant security guard in a museum, and when the artifact, that is DNA, is damaged, the guard can either put up a barrier to fix it or decide to remove it altogether if it's beyond repair. In cancer, it's as if the security guard's gone missing, so the damage can accumulate unchecked and it can lead to chaos within the museum. A growing number of other genes involved in DNA damage sensing, repair, and chromosomal segregation during mitosis are also found to be lost or mutated in various cancers. These genes, often referred to as caretaker genes, are labeled as tumor suppressors because their normal function suppresses tumor formation by maintaining genomic stability. When these genes are lost or dysfunctional, it allows for genome instability and variability, generating mutant cells with selective advantages. Selective advantages in this context refer to traits that allow cancer cells to grow and divide more rapidly than normal cells. Interestingly, recent evidence suggests that apoptosis may also contribute to this genomic instability. When cells undergo apoptosis, their DNA is fragmented into small pieces. If these apoptotic cell bodies are phagocytosed by neighboring cells, the DNA fragments can be incorporated in the genome of the engulfing cell, potentially introducing new genetic variations. This process can diversify the genetic makeup of tumor cells and provide further opportunities for acquiring mutations that drive cancer progression. As an analogy, think of apoptosis as a janitor cleaning up a room. Normally, the janitor would sweep up the debris and throw it away. But in cancer cells, imagine if the janitor starts picking up pieces of a broken jigsaw puzzle and randomly adds them to other puzzles. And this random addition can change the puzzle's picture entirely, as just a few DNA fragments can introduce new mutations. Genomic instability is distinct from the 6 hallmark capabilities associated with the tumor phenotype and tumor physiology. It serves as an enabling characteristic, allowing evolving populations of pre-malignant cells to reach these 6 biological endpoints. This instability provides the necessary mutability for cancer cells to acquire the capabilities required for tumor progression and malignancy. Essentially, genome instability accelerates the rate at which cancer cells can accumulate the mutations which are needed to survive, grow, and spread. So think of it as a house on a shaky foundation. While the house might stand, it'll constantly shift and change, making it easier for new mutations or extensions to be added, and this unstable foundation is what allows the house to grow in unpredictable ways, just as genome instability allows cancer to progress. So as we continue to unravel the complexities of cancer biology, it is crucial to adopt a holistic view that considers not only the genetic mutations within tumor cells, but also the tumor microenvironment. The tumor microenvironment includes the surrounding cells, blood vessels, immune cells, and signaling molecules that interact within the tumor. These interactions play a significant role in cancer progression and response to treatment. Imagine a future where cancer is not the enigmatic adversary it is today, but a well-charted territory where every signaling pathway and every genetic mutation is understood and targeted with precision. This future holds promise not just for effective treatments, but for preemptive strategies that could stop cancer in its tracks before it even begins to take hold. Precision medicine aims to tailor the treatment uh, towards the individual characteristics of each patient cancer, considering their genetic profile, lifestyle, and environment. So, if you think of it, cancer like a vast forest, we only have a vague map for a few items and where they are, and the future holds the promise of a detailed GPS system that can guide us and avoid ineffective treatments and find the best parts, the best or the most effective therapies to the cure. And as we move forward and hold on to this vision, the fight against cancer is obviously far from over, but with each discovery and technological advancement, we step closer to a world where it's possible. And with this episode, we have brought to an end the first six hallmarks of cancer that were described in the first Hallmarks of Cancer paper published in 2000. The next edition we'll be talking about is Hallmarks of Cancer, published by the very same researchers in 2011. So, see you then.